equally participate in this thing. So my wholehearted thanks to the team of the Young Professional Forum for taking this initiative when our country is passing through such a bad phase of COVID-19 pandemic, where everything has come to a grinding halt, including the academic activities. Our students are suffering for that, and this is really a thing support which everybody should wholeheartedly support. Thank you very much.
Thank you, sir. Dr. Ankit.
Thanks, Aikaj. It's my pleasure to introduce Neelam Marwaha ma'am. Most of us has been taught by madam at various different platforms, academic platform. She's an excellent teacher and she has taught me also. It's my great pleasure to introduce ma'am here at YPF Academy webinar. So she is convener of MD Transfer Medicine program in India. She is chairperson of TRG group in NACO. She is chairperson of National Hemovigilance program. If I'm not wrong, I think she was the one who took initiative to start hemovigilance program in India, which has been appreciated all over the world at various international forums. She is regional director of Southeast Asia at ISBT. With this, I hand over to Madam. Would you like to say some few words for IPF Academy?
Yeah, so thank you so much for your very kind and generous introduction, and very good afternoon to everybody. And happy to see so many people already, students, participants logging in. And at the outset, I just want to share that when we founded the Indian Society of Transfusion Medicine, it's almost going to be 10 years. You know, our dream was to have a forum where we could promote science, academics, education, training, give fellowship programs to our students. And, you know, we were having conferences and very good quality conferences, but that was mainly some scientific exchange. And there was a very big gap or lacuna which everybody was feeling, and that our students were not getting that kind of opportunity for education, for training, for coming forward with the problems that they felt and we also felt were very genuine and needed to be addressed. So keeping that in mind, I think this Young Professional Forum, which is really an academic initiative of the ISTM, for me, it's like, you know, an academic dream which has come true. And I really appreciate the dynamic leadership of Professor Debashsish Gupta, President-elect of the ISTM, along with the team of very enthusiastic, brilliant and dedicated young members, Dr. Abu Bakr, Ekaj, Dr. Ankit Mathur, Dr. Puneet Jain, and Dr. Rizwan, Dr. Subro. So many young, brilliant people and who are representing the entire young community. And they have come forward with an excellent program. And I think, you know, covering the topics which are in the MD curriculum, DNB curriculum is very important because we needed to address this gap in education. And I have also seen that there are opportunities for free paper presentation, opportunities for journal club presentation. So along with learning, there is also a lot of emphasis on improving the oratory skills, confidence building, trust building. So I really congratulate all of you. And I appreciate the excellent efforts which have been taken by Ekaj on behalf of the forum and the entire fraternity to coordinate, because coordination is also a very big thing, especially in these challenging times, to get people on board. And before, if you give me a minute, I just want to give bit of advice, I should say, or a suggestion. I should not say advice, not a good word, but a suggestion for the students. that whenever they are listening to a seminar, I think you should always keep in mind three objectives, or you should look at it from three angles. One is, will this topic which is being presented increase my understanding of my specialty? Will it anyway help to improve my day-to-day practice of transfusion medicine? And does it open up new research frontiers for transfusion medicine? So look at the topic, view the topic with these questions in your mind. This will not only help you increase your attention, but also at the same time, it will keep you more involved and more focused in the specialty. So with these few words, congratulate Professor Debashsish Gupta with his team of young, brilliant people. And I'm so happy to share that Dr. Vijay Kumawat, who is the opening, I would say, academic batsman of the ISDM academic initiative, is also an alumnus of PGI Chandigarh, and I really know and that he's going to make a very meticulous presentation. And thank you so much, all of you, for allowing me to share a few thoughts with you. Thank you so much.
Thank you, ma'am. Before we actually go on to the main event of inviting Dr. Vijay Kumawat, I'll take just a small moment to introduce everybody who's been working behind the scene. And we are, just a group of people who thought that, yes, we wanted to do something for the transfusion medicine fraternity. And let me start by saying this, we are all volunteers. We do not hold any post. We are just volunteers who have volunteered that, yes, we will work without any money, and we will just further the academics and will percolate it down to the grassroots level. I take extreme pleasure in introducing my colleagues in this endeavor, Dr. Abu Bakr Mohammed Rafi. He's Assistant Professor, IHVT, Jubilee Mission Medical College Research Institute, Kerala. Rafi, if you could just say hi to everybody, we would love it. Rafi, are you there? So we might be experiencing some issues. We are these 3 months, the first 3 months, as under the guidance of Dr. Debashsish, this is just a trial run of all the good things that are going to come January onwards. We'll try to remove all the teething issues that we have, all the software problems we have, and we'll try to increase the participation and get feedback. Second person of our team, I will move in alphabetical. This is me. I wouldn't say much about me. Dr. Puneet, he's a consultant at. I would request Dr. Puneet to introduce himself.
Hello, good afternoon, everyone. Good afternoon, ma'am and sir. So I am Dr. Puneet Jain, and I am currently working at Apollo Hospital, Navi Mumbai, as the consultant and in charge of the blood bank. And I am happy to be a volunteer in this initiative. And Aikach, you can introduce our rest of the team.
Thank you. Dr. Rizwan is with us. Dr. Rizwan. Dr. Rizwan is presently consultant in neophyresis and cell processing with Tata Medical Center, Kolkata. He would be joining us soon. Dr. Ankit Mathur. Sir, please.
Hi, everybody. I'm Ankit Mathur, and I work at TTK Blood Bank, Bangalore, and happy to be a part of YPF.
Dr. Suvro, he could not join us as of now due to some pending inspection at his place. He's consultant transfusion medicine, Tata Medical Center, Kolkata. New members to our team, Dr. Anisha Navkutkar. Dr. Anisha is consultant, Department of Transfusion Medicine, Tata Memorial Hospital, HVNI, Mumbai. Dr. Anisha, please, we would love a shout out from you. You're muted, Dr. Anisha.
everyone, and thank you for inviting me here. Also, thank you all that this forum is a very nice initiative for all our youngsters, and we wish a very all the best for all our participants as well. Thank you.
Our next volunteer is Dr. Soumya. Dr. Soumya Das is Assistant Professor, Faculty in Charge, Department of Transfusion Medicine, AIIMS, Nagpur. Dr. Soumya, please say just a shout out to know that you're there.
Yeah, I'm there. Hi, everyone. I'm Dr. Soumya Das, as already Arkesh told, and I'm happy to be a part of this initiative. And I'm volunteering for all the scientific events which are going to happen in the near future for the academic fest. Thank you.
And the next volunteer we have is Dr. Trupti. Dr. Trupti Barot, are you with us? I am Dr. Trupti. Please, we would want a shout out from you.
Hi everyone, myself Dr. Trupti Barot, working as a transfer medicine specialist in Ratna Blood Center. I am happy to be a part of YPF, and all thanks to everyone for supporting us. And we are great. We have best wishes to all and all academic team.
Dr. Rizwan is back with us. Dr. Rizwan.
Hello everybody. It's wonderful to be part of the ISTM and in the time to come. I have with me Dr. Shubham.
So hi everyone. So I'm here with Neelam. So best of luck for our initiative and happy academics.
And so, without any more further ado, we would like to invite Dr. Rizwan to please take over and introduce our speaker.
Just one announcement before Dr. Rizwan takes up. For this webinar, we'll be issuing the certificate of participation through YPF ISTM, and the link for registering for certificate is available in the chat box. So I would request everyone to please fill up the Google form and submit it so that we are able to issue the certificate. Thank you. Rizwan, you may take over.
Thank you so much. It is a pleasure to introduce Dr. Vijay Kumawat. I had a brief interaction with him, and it was great to know a polite person like him, who is presently also the Associate Professor at the Department of Transfusion Medicine and Hematology at NIMHANS, Bangalore. He completed his MD in Transfusion Medicine from PGI Chandigarh and subsequently did a PG DMLS from Symbiosis International University. He's also earned a diploma in tissue banking from the National University Hospital in Singapore. And his previous professional experiences are that of a senior resident in PGI, assistant professor at GMC Nalhar in Haryana, and a head and consultant at Citizen Hospital, Hyderabad. He is written several publications in international and national journals, and a reviewer for several noted journals, as well as societies working in transfusion medicine in India and abroad. So it's a great pleasure. We are looking forward to hear from you.
Yeah, thank you, Dr. Rizwan, for the kind introduction. Am I audible, Dr. Ekaj?
Yes, sir, you are audible. We would request you to please start sharing your screen.
Okay, just a minute. So, is it visible, moving?
Yes, sir. Yes, sir.
Yeah, so should we start?
Yes, sir.
Okay, good afternoon all. And good afternoon, Marwaha ma'am and Dr. Sharma and Dr. Debashsish sir. It's my pleasure to begin this academic session. So the topic which is allotted to me is red cell metabolism and anticoagulant. So we, as a blood banker, is always fascinated with the red cell and other component also. But red cell, because blood looks red, so we are all have fascinated thing for the red color. But it's interesting that red cell is attracting other investigators, particularly the basic science researchers. So RBC, by virtue, is the most abundant host cell in human body. So we can say almost 2.5 into 10 to the power 13 out of 3 into 10 to the power 13 cells in human body. This is just a rough estimate. Earlier, RBC was regarded as just a mere oxygen carrier, but now with the upcoming research and data, we can say that it is a key contributor to systematic metabolic homeostasis also. Currently, till now, around 2,800 proteins have been identified. So we can see it's not a simple cell, it's a little complex structure also. And as we were talking about the research opportunity in the transfusion field, so the omics is the new generation research which has come up in the last decade. For that, RBC represents a simplified model for eukaryotic cell because it has a stable proteome, because we know that red cell doesn't have much of the de novo protein synthesis capacity. So if we talk about metabolism, it's basically first thing is the generation of energy to maintain all vital cell function. It can be either it is intravascular lifespan or in vitro storage in the blood center. So mainly it is focused for maintenance of glycolysis, maintenance of electrolyte gradient, synthesis of glutathione and other few metabolites which red cell is able to do, like purine and pyrimidine metabolism. The important function of the red cell is maintenance of hemoglobin iron in its functional reduced ferrous state. Protection of metabolic enzymes because it carry oxygen and there is potential for oxidative injury to the cell and the hemoglobin. And the preservation of membrane phospholipid asymmetry so that it can retain its shape, which is quite important for its prime function of oxygen transport, which happens through the capillaries, which is almost less than half of the size of red cell at capillary level. So we go with the key highlights of RBC metabolism. So there is no oxidative pathway or Krebs cycle or tricyclic acid cycle. So the primary mechanism in the red cell is anaerobic. So which is called glycolysis or Embeden-Meyerhof pathway, which was discovered somewhere in 1930. And erythrocyte possess a unique glycolytic bypass pathway, which is mainly to produce 2,3-biphosphoglycerate. So this is called the rapid Leu-Behring shunt. If we see in any cell, in any nucleated cell, the metabolic regulation is dependent on the protein synthesis, which is mainly regulated by increasing or decreasing DNA transcription or translation of messenger RNA. Here we can see the pictorial that nucleus will give command and the various organelle will function. But a red cell, because of its prime function to carry oxygen, so it is full of hemoglobin. So it sacrificed most of its organelle, including the nucleus itself. So red cell do not have these options. So this is how red cell metabolism is going to differ from a nucleated normal cell, which we say, where the red cell is being a specialized cell for a very prime function in human body. So we start with the glycolysis. It is the same glycolysis which we have been reading in our biochemistry book during our UG days. And it is most phylogenetically old biochemical reaction. Still the similar step happens in a most primitive state like in the bacteria. And glucose is the main substrate for this reaction. So we have discussed that despite it is full of the oxygen, but still the red cell cannot use that oxygen for its own use for energy production. So it's mainly all anaerobic. So this is the one pathway I am going to show it in the break. So this pathway starts from the glucose to the generation of phosphate isomerase. So now we move to second part of this glycolysis, where this rapid Leu-Behring shunt also comes into the picture. So it starts with the glyceraldehyde 3-phosphate and it stops at 1,3-diphosphoglycerate with the production of NADH. So here we have this shunt. So production of large quantity of 2,3-DPG, it is the unique feature of glycolysis in the red cell with this shunt. So here the shunt takes and it goes producing 2,3-biphosphoglysate, which happens with the help of one single enzyme, which acts on the both way. It can act as a mutase or it can act as a phosphatase depending on the pH. So at higher pH, the enzyme acts as a mutase and it generates lots of 2,3-diphosphoglycerate or biphosphoglycerate. And at the lower pH, it acts only phosphatase. So it diverts all 2,3-DPG again to the glycolysis cycle. So this 2,3-DPG shunt, which provide a very important molecule for the red cell, it happens at the expense of ATP, because with one glucose, we know that it generates 2-diphosphoglycerate. So with this total process, red cell loses the 2 ATP and gain the 2,3-biphosphoglyscerate. So it has implication in the storage system, because high pH where this enzyme acts as a mutase, it can shut down the complete ATP production, and mutase will be very highly active and it keep producing 2,3-diphosphoglycerate. Or if it is low pH, then it can completely deplete the red cell of 2,3-DPG and there can be a sudden burst of ATP production within the red cell. So we have to understand this. So pH of the RBC, particularly during storage, is very important, because if we see above 7.2 pH, this bifunctional enzyme, it will convert almost all 1,3-DPG into 2,3-DPG and depriving cell of new ATP. Below 6.4 pH, if it goes up to that level, then activities of almost all enzyme of the glycolysis, particularly the rate limiting enzyme, which is hexokinase, phosphofructokinase, will be very too low to support ATP production, and there will be no source of energy for the red cell. So that's what when venous blood, which is approximately a pH of 7.35, it is being mixed with the anticoagulant nutrient solution, which is usually depend on the anticoagulant solution. pH range is somewhere around 5 to 5.6, which results in pH of 7.05 in the mixture. It is on the day 0 when the bag is completely filled. So now the next part of glycolysis, where the final step of this glucose breakdown happens and the first formation of pyruvate, which ultimately turns into the lactate. So here the implication for this metabolic pathway is, because lactic acid, when red cell is in the storage medium or in the blood bag, it accumulates during the red cell storage, because blood cannot wash out this lactic acid. And accumulation of lactic acid is the principal reason, because for that only we observe the glycolysis rate coming down as the storage duration progresses of red cell. So this was one of the primary metabolism which happens in the red cell. I'll come with the other metabolic pathways also. But first, to understand all metabolic pathway, we should go briefly towards regulation of metabolism in the red cell. So as we have seen that red cell is devoid of nucleus, there is no DNA transcription, no RNA translation happening in the mature RBC. So the metabolism, red cell metabolism, is principally regulated by feedback mechanism. And still, despite of extensive studies, our understanding of regulation of metabolism is still incomplete. So lots of research is still going on, and lots of research, so there is a huge potential for the research as a transfusion specialist for us also to discover all those metabolic pathways which are still not known, unknown, and we don't have many answers. So we are all aware of this oxygen dissociation curve, which is having sigmoid shape, and we all know that what is the right shift, what is the left shift. So it is mainly controlled by the DPG level concentration, the temperature, and the pH, or mainly the hydrogen ion concentration. So if we see the basic oxygen molecule structure, so in relaxed stage where oxygen is bound to the iron, and in the tense stage where 2,3-DPG bind to the hemoglobin molecule and releases the oxygen. So binding to hemoglobin, this 2,3-DPG is very important, because if we see in the relaxed stage where oxygen is binding to the hemoglobin molecule, approximately 80% of the 2,3-DPG is free. And the same time when 2,3-DPG bind to the hemoglobin, it decreases the oxygen affinity, hemoglobin releases the oxygen, and almost 80% 2,3-DPG in this form is remain binded to hemoglobin. So another regulatory mechanism which is quite important, it happens through the red cell membrane. So we can see in a red cell membrane, there is a lipid bilayer, which is common in most of the cells. There is a membrane skeleton below the red cell membrane, which is mainly made of spectrin, alpha and beta chains. Then there are various intermediary proteins which binds this membrane skeleton to lipid phospho bilayer, so which are mainly like protein 4.1, anion exchanger, band 3, band 4.2, and ankyrin. So an intact membrane skeleton is crucial for mechanical and functional integrity of the blood cell, because it determines the viscoelastic properties. So this allow to withstand the shear forces of circulation when blood is flowing and red cell is flowing with the circulation. And this is also important when this red cell need to pass through the capillaries to have effective oxygenation. So we know that lipid bilayer, there are the proteins. So always lipid bilayer of red cell or any cell, it is asymmetrical, and this asymmetry is quite important. So the outer layer usually mainly dominated by phosphatidylcholine and sphingomyelin, and while lower layer, it is mainly by phosphatidylinositol, phosphatidylethanolamine, and phosphatidylserine. And this phospholipid asymmetry is maintained by ATP-dependent flippage. And this flippage activity decreases during the storage. So that allows this phosphatidylserine to expose on the outer layer. And there it can be a marker for red cell sensitization, and red cell can be removed from the circulation when it passes through the spleen. So if we see the band 3 anion exchanger, which can be there as a tetramer in 60% total mass, and it present as a dimer in the 60% and tetramer as a 40%. So this band 3 is having three main domain. One is membrane spanning domain, another is short terminal C cytoplasmic domain, where this carbonic anhydrase 2 keep binding, and another is a large N terminal, which binds to lots of molecules, which includes deoxyhemoglobin, aldolase, glucose 3-phosphate dehydrogenase, and hemochrome. So all will have a role in regulation of red cell metabolism. So if we see when hemoglobin is binding to the oxygen, the level of 2,3-DPG, free 2,3-DPG is the minimum, somewhere around 0.5 millimole within the red cell. When red cell bind to the hemoglobin, the free 2,3-DPG level increases to 5.9 millimole, which is the maximum thing. So when hemoglobin bind to the oxygen, 2,3-DPG is released from the hemoglobin molecule, and this free 2,3-DPG bind to the band 3 complex, and which results in three major activity. One is it inhibits the binding of protein 4.1 to band 3. It releases spectrin from the membrane skeleton, and it interferes with the spectrin actin protein 4.1 association. So mainly this 2,3-DPG is releasing membrane cytoskeleton from the phospholipid bilayer, and the flexibility of red cell is increasing. So this in turn facilitates gaseous exchange, so red cell can slip through the narrow capillaries and splenic sinusoids. So if we see another mechanism which is the part of red cell metabolism, an important portion, which is the release of oxygen by red cell as tissue oxygen level. Because movement of erythrocyte through the tissue and lung capillaries is very rapid. So transit time remains between 0.3 to 1 second. So it is on an average, it is less than half a second. So what happens here, the band 3 macro complex, which provide a short path for oxygen, carbon dioxide, protein, bicarbonate movement. So what happens when the red cell reaches the tissue level, which is in need of the oxygen. So there is a carbon dioxide abundance there. So carbon dioxide is taken care within the red cell through the help of this band 3 complex. This carbon dioxide is converted to bicarbonate ion and with the help of carbonic anhydrase enzyme. So one bicarbonate ion and one proton is basically carbonic acid is being generated, which breaks down into bicarbonate ion and the hydrogen ion. This bicarbonate ion gets out of the red cell in exchange of one chloride ion, and the same proton is released. And as we have seen in the oxygen dissociation curve, the protein shift the oxygen curve to proton, the hydrogen ion shift the oxygen dissociation curve to the right side. So because of this hydrogen ion, which is called the Bohr effect, the oxygen is released from the hemoglobin, which is again with this band 3 complex is released at the tissue level. The water required for this reaction is supported by aquaporin receptors, which are there in the red cell membrane. So now we come to one of the again important pathway, which is pentose or hexose monophosphate shunt. So which is the important for generation of NADH or the reduced form. So in this, glucose 6-phosphate is dehydrogenated and ultimately resulting in the formation of ribulose 5-phosphate and producing the molecules of NADPH. So this final ribulose 5-phosphate can have two fate. It can be converted through various chains of reaction to fructose 6-phosphate or to glyceraldehyde 3-phosphate. And these two molecules again can enter into the glycolysis. So it saves the glucose molecule. Which glucose molecule it is started? Again, that glucose molecule enters in the glycolysis and generates the ATP. Again, another important aspect of this pentose pathway, this ribulose 5-phosphate converted into the ribose 5-phosphate. These are just an isomeric reaction, and it joins the phosphoribosyl pyrophosphate, and through this reaction, it generates adenine nucleotide for the red cell. So the importance of this pathway, it serves as a source of NADPH. So mainly it maintains glutathione in its reduced form. So this reduced glutathione is important for elimination of peroxide, protection of protein sulfidyl group, again from the high reactive oxygen species, limitation of lipid peroxidation and detoxification process. So how does it happen? I have just taken a simple example for hemoglobin, where like this NADH get oxidized, and the enzyme is NADH cytochrome B5 methemoglobin reductase, which help to maintain iron into the reduced form. So iron should remain in the ferrous form so that it can bind to the oxygen. So it get oxidized to the NAD, and the same hydrogen is being used to reduce methemoglobin to hemoglobin. Again, because of oxidation reaction, this can be converted to methemoglobin, and again with the pentose phosphate pathway, this NAD will be converted to NADH. So this is how the red cell is maintaining and preventing oxygen and other protein from the oxidative damage. So how this is balanced, that is quite important because regulation, like when should red cell go for glycolysis, when should red cell go for the pentose shunt. So there is a regulatory mechanism, which is again dependent, what we have till now, the information is dependent on band 3 complex, which is hereby in diagram, it is shown as B3P. That's what I have given the initial overview of membrane cytoskeleton. So what happens in high oxygen state, where the red cell is carrying the maximum amount of the oxygen? At this time, most of the glycolytic enzymes like phosphofructokinase, aldolase, G3 phosphate dehydrogenase, and LDH get bind to band 3 complex, and So if enzymes are not working, glycolysis will not definitely work, and that time the shunt will work. This hexose monophosphate shunt will work, and it will generate more of NADPH, so that the oxidative injury can be prevented, which we expect at the high oxygen level to the red cell. Inversely, when red cell is at low oxygen state, when the hemoglobin has released most of the oxygen to the tissue level, that time what happens? Deoxygenated hemoglobin, what we have seen, that deoxyhemoglobin also bind to the band 3. So deoxyhemoglobin bind to the band 3 complex and releases the enzyme which are involved in glycolysis. So at this stage, glycolysis is more preferred metabolic pathway than your shunt. So now red cell is involved in a few of the synthetic processes, which few we have seen. So mostly it is achieved by the salvage pathways. So it try to retain the or recycle the molecules. So main, like we have seen, adenine is regenerated, then again through phosphoribosyl pyrophosphate, it can be converted to AMP. Guanine nucleotide also being generated through the similar reaction. It can produce the reduced glutathione, it produce NAD, nicotinamide adenine dinucleotide, and then some amount of S-adenosyl L-methionine also. So if we see the adenine nucleoside metabolism within the red cell, so the first reaction, ATP and AMP, they usually remain in a balanced position to 2-ADP. So because of this reaction, when the normal glycolysis is happening, AMP concentration remains low and ATP keep generating. When glycolysis slows down, which usually happens during the storage when the level temperature is low, so that also plays a role to slow down the glycolysis. And at the end stage of the red cell storage where pH level goes down, that also contribute to reduce rate of glycolysis. So level of ADP start increasing, and then it keep converting to AMP, and AMP in turn deaminated, and then it comes out of total red cell pool. Then it cannot be reutilized. So that is the importance. If adenine is added to the anticoagulant or to the additive solution, the red cell is better preserved, so that the AMP is available to produce ADP, which in turn can help in generating ATP. Same guanine nucleotides. So red cell contain and turn over guanine nucleotide. So this play a very important role in signal transduction of the membrane, particularly for the secretion of local vasodilators like cyclic AMP and ATP. So high concentration of GTP inhibit red cell transglutaminase, but with aging, the level of GTP goes slow. So this transglutaminase facilitates removal of senescent red cell by binding them to the fibrin clots. So another important part of red cell metabolism is the sodium potassium channel metabolism, which is one of the important anion channel. There are other anion channels also, but we'll focus on the sodium potassium channel. So what happens, the sodium potassium channel, which is there in the red cell membrane, in the initial stage, it binds to the 3 sodium and 1 ATP molecule. When ATP get dephosphorylated, like it releases 1 phosphate molecule, which generates the energy, the shape of sodium potassium ion changes, which facilitate sodium ion to go out from the cytoplasm and enter the pump. And the final reaction is 3 sodium are released and 2 potassium are being taken inside the cell, and release of phosphate allow 2 potassium cell to enter into the, so this is how red cell keep maintaining the sodium potassium gradient. So, but the important part from the storage point of view, this sodium potassium ATPase activity, which is the enzyme required here to act, it is highly sensitive to change in the temperature, and it hardly function at 4 degrees Celsius, which is the storage temperature of in vitro red cell storage. So that's why this sodium potassium channel doesn't work at all during the storage. So sodium diffuses into the cell and potassium leaks out until a new equilibrium is reached, depend on the concentration. So that's how we can see that more potassium in the plasma, stored blood plasma, which can be deleterious to particular patient population, which can be increased by few of modification of the red cell like irradiation. So it can be deleterious for the high potassium level for a neonate or for a patient who is in critical care where the already electrolyte imbalance is there. So that thing we have to keep in mind. So now I'll move to the next portion, which is anticoagulant. But basically, it is good to say anticoagulant and the nutrient and preservative solution, because it is not only doing anticoagulation, it is providing nutrients which are required for the red cell metabolism, and it is helping to maintain better preservation of the red cell, which is still we are trying to update and put the new knowledge, new investigation, new research, and it's keep updating every second year. So the most commonly available anticoagulants which we use day to day is ACD, which is like commonly we know acid citrate dextrose, where the red cell storage life is 21 days. The CPD, again the life is 21 days. We have CP2D, double dextrose population. The shelf life is again 21 days, and the CPDA, which is commonly used, is 35 days is the shelf life. If we see the role of individual component, which we all know that citrate helps to chelate calcium and prevent clotting. Phosphate maintain the pH during the storage, and it is helping to maintain adequate level of 2,3 DPG. Dextrose is the substrate for the ATP production, and adenine is helping in production of ATP, which had helped us to extend the shelf life from the CPD ACD era, where the shelf life was 21 days, to 35 days, and later on native solution helped us to move to 42 days. So if we go how this concept started of red cell anticoagulation, so somewhere it is starting from the 1890. Before, so scientists were looking for various methods where they can use a effective anticoagulant which is non-toxic to the human body. So the Roche and Turner, they developed the first red cell storage solution, which was a simple mixture of citrate and glucose. This is their publication, which was published somewhere in 1915. So the essence of the abstract was they want to use a state of suspended animation for the red cell, which is some way now with the current understanding, we can say half of it was true, half of it was not true, which is like keep improving with the more and more research coming up. But the interesting thing for this was when the Roche and Turner used the glucose along with the citrate, they also even don't know that glucose was the substrate and it is metabolized by the red cell through glycolysis, because glycolysis was somewhere discovered in 1930, though the initial work was going on how energy being generated, but so they were just looking for some of the compound which can give stability to the red cell. So this is the initial earliest blood collection device, which was developed by Oswald H. Robertson, and he is also given credit to establish one formal blood bank in the USA. So it was with the help of Winchester bottles, which was around 1.5 liter capacity, which is filled with the isodextrose. Over that, a layer of isocitrate was added, somewhere in 350 ml quantity. And in this bottle, 500 ml blood was collected from the donors with the help of needle and rubber connection and glass inlet tubes and suction pump, we can see. So then the scientists were trying to improve the blood quality, so focus was there. So the Lottet and Mollison, they first introduced the disodium citrate glucose as a mixture for blood preservative, because with the initial citrate and glucose mixture, problem was with the autoclaving. Whenever you autoclave with the sugar, it gets caramelized. So they have started using disodium citrate. So this was having the advantage that the entire solution can be autoclaved altogether, because initially before this storage medium, the citrate was separately autoclaved, then glucose was separately autoclaved, and then they put under aseptic conditions, but still there were chances of bacterial contamination to this solution. So the finally from the same group, the Mollison and Lotted, they have come up with the citric acid, sodium citrate, and glucose mixture for the blood cell storage somewhere in 1943. Before that, the MRC solution, which is commonly used, this is Medical Research Council solution, because it was given in a army memorandum that this is the formula you have to use for blood preservation. So it is fondly known as MRC solution. So they have come up with the CPD solution. So in this CPD solution, red cells were better stored, and this whole mixture can be autoclaved with production of little caramelization. And the transfusion with this red cell or whole blood stored in this medium was uneventful, and amount of methemoglobin formed with this blood stored was not different from this MRC solution. So from historic point of view, it's good to know that what was the MRC solution for the young PGs. So finally, they have shown, because now we got the citric acid, so it should be at the low pH. We got one constant at citrate, and the glucose was there. Then again, researchers were fascinated to improve the red cell quality. So so the phosphate was studied extensively by the Gibson and his team. So he proposed this percentage of anticoagulant solution. So he had shown that when we use CPD, citrate phosphate dextrose, in place of ACD solution, the mean survival for at 28 days for ACD solution was 76% in comparison to 76% at CPD solution. So they have shown that if we add the phosphate to the current anticoagulant solution, the red cell preservation is better. So with the same group investigation, if we see, so they have compared the plasma level between both solutions. So in this, we can see the phosphate levels of inorganic phosphate is always higher than the ACD solution. And the same group, the compare intracellular phosphate concentration. So if we see the phosphate concentration with ACD solution is increases up to 2 weeks, then it come to a plateau phase. But in CPD solution, the intracellular phosphate solution is constantly maintained up to 4 weeks. So that's how the CPD solution is helping better preservance of the red cell. So the later on phosphate was added and CPD solution was continued. So the same group has checked the relationship between the early recovery survival. So which was here we can see with the graphs, it is better in the CPD solution. So this Orellana group in somewhere in 1969, they again continued the Gibson work. So by this time, it was the understanding that addition of phosphate to ACD improved the red cell survival. Still, we were behind. So in the same year, somewhere in 69, Shield, they have shown the effect of adenine for improved autologous and homologous transfusion. So this adenine was added, and this resulted in nowhere increased erythrocyte breakdown, and the red cell survival was better, 70% at that time, post-transfusion survival, even after 42 days of the storage. So addition of adenine, which we have seen how adenine is going to help to improve the red cell metabolism, had helped to extend the shelf life up to 35 days. So this was the breakthrough study, which has proven, because earlier the researchers, scientists were not convinced because they were not sure if adenine breakdown molecule, how it is going to behave in the patient population when it is going to be transfused. So this research group has shown that red blood cell stored in modified CPD with adenine as a whole blood for 35 days and red cell concentrate at 28 days were compatible to the red cell stored in CPD and ACD. And this concentrate showed satisfactory survivability of the red cell and no deleterious adverse reaction to the patient population. So this is from my side for today. Hope everyone is awake so we can discuss further things. Dr. Ekaj?
Dr. Kumawat, thank you so much for such a wonderful, detailed, comprehensive presentation. I would request Dr. Rafi to please initiate the discussion. All the chairpersons and the speaker is open for questions. If you have any questions, you can unmute yourself and ask the question, or you can type it in the chat and then you can ask it. Both ways.
Thank you, Dr. Vijay sir, for that elaborate talk. It was very much elaborate for especially for the students, for the PG students. We have around 155 participants as of now. Any student, any participant, if you have any queries, you can either unmute yourself and ask, or you can also type in the chat box. You can raise your hands also. I would also like to remind that all participants, we are also issuing certificates. So there is a Google form, which is there in the chat box. Please do fill it and we'll be sending certificates to you.
Hello, can you hear me?
Yes, go ahead.
Yeah, hi, Dr. Vijay. I'm Dr. Somnath.
Yeah, hi, sir.
Yeah, hi, how are you? Yeah, it's a good, fine. Very nice presentation and very elaborative presentations. Thank you for these presentations. But I would like to you just highlight few developments of new preservatives. Can you please highlight these few recent developments of any preservative additive solutions?
Additive solution was not part of my talk, so I deliberately left it.
Okay.
Because it was up to anticoagulant only. So
I'm sorry. I thought that because when you were talking about the up to CPDA, then I thought you'd progress further. That what is the latest?
No, sir. Because 30 minutes was given from the forum, so it would have been injustice if I try to cover a lot thing. So maybe later on we can do somewhere next time additive solutions.
Sure. Thank you.
Yeah. So there is one question from here. I can see someone asked for this why only ACD is being used for apheresis. Hello.
Yes, Dr. Vijay, you can answer to that. For apheresis, why only ACD is being used?
Yeah. So frankly speaking, I don't know answer, but still I'll try to explain what I got through and till now I know. So the addition of phosphate to the platelet, it is a still debatable thing, because you see only for apheresis platelet we are using the ACD, but if you talk about the preparation of platelet from the whole blood, the CPDA solution is there or CPD is there. So if we pool it, the phosphate will be there. So the phosphate works in two ways. One thing is phosphate promotes the glycolysis. It speed up the glycolysis. So the earlier concern with the use of phosphate in the specific platelet product, because in the red cell product in the whole blood we cannot avoid, because we have seen that phosphate is helping to better maintenance of red cell. So for platelet, it was thought that phosphate will promote the glycolysis and it will rapidly deplete the glucose which is available within the limited amount of platelet when we collect suspended in plasma. Later on, when the platelet metabolism get explored, now we know that during storage, the main substrate for platelet metabolism is free fatty acid, and if provided in additive solution, then acetate is the better choice. So when we move to platelet additive solution, acetate is the constant thing. But phosphate also acts as a buffer also. Okay. So in few additive solution, if you see, the phosphate has been tried. I think in some past 3 solution, the phosphate was added, and it was shown that there is no difference. It maintains the platelet viability in the same way. Okay. So that's what I have. If anyone in the forum has few more addition, always welcome. Dr. Linda.
Dr. Linda Joan, does that answer your question?
Yeah, any other queries? I think if anyone has an answer, like anything more to add to that?
I think it seems to be a very plausible answer which Dr. Vijay has given. And so that could be probably the underlying reason why we have a different solution. So I don't think there are any more questions. We don't see any more. So, but one thing, yes, I want to actually compliment the people who have selected this topic, because generally we used to study or teach red cell metabolism separately, the anticoagulants separately. But here combining the two and trying to correlate why you need to understand the metabolism and what is the implication on the storage and the various storage anticoagulant preservative solutions you have. So I thought this was a very good way of having a presentation. So if is Dr. Debashsish there? Would he like to add anything?
Oh, I think presentation was quite comprehensive.
Yes.
And he has very well taken both the thing together, the red cell metabolism with the along with the storage thing. So I actually compliment Dr. Vijay for this excellent presentation. What is my only one thing is that since these particular topics are being circulated to all the our participants earlier, so they can come prepared on what particular point they really want to understand from the presenter in the next coming weeks whenever they attend to this step of a webinar.
Now, I just want to ask that supposing, later on there are some questions and because these are going to be time bound, but Dr. Ekaj, is there any mechanism of addressing that? It's possible some students may have some questions later on, or when they are going through a presentation or a textbook, something might just occur to them. So how do we have any
Yes, ma'am.
plans of addressing?
Basically, we will encourage, as Dr. Debashsish already said, we would encourage all the participants to actually read something about the topic before coming. And in their subsequent study, if they have a question, please email those questions to YPF. We will coordinate and connect the speaker with those students, and maybe they can have one-to-one interactions with each other. Because we are involved not only for this webinar, but for the entire duration of next 1 year. Any questions that you have, we are there for you.
I think these queries which are being raised now by both by Dr. Neelam or other participants, whoever raises queries, we should take a close introspection about this thing, that how we can address this problem issues. Okay, so in the coming days we can sort out all the things that regarding the timing factors, all other small things which are coming into the discussion.
Right, sir.
We are also recording this session. At the end, every week we'll be uploading it also.
No, that's wonderful. And I think, as the inaugural webinar, I must say it is excellent. There were no glitches, no hitches, nothing. I think it was very smoothly done. And hats off Dr. Debashsish, Dr. Ekaj, and the other members of the forum, and Dr. Vijay Kumawat. for a very, very lucid and excellent presentation. I really enjoyed the whole
Yes,
presentation.
one more thing that there are more than 155 participants in this
Yes.
inaugural session. So I hope that in the coming weeks, our number of participants will go up. And there should be word of mouth publication. This thing should be there so that people can come and participate more in the coming weeks.
I think we have 155 people to advertise, which is a very big number.
Yes.
Yes.
Ma'am, we have two messages before we go. Dr. Puneet, your message.
Yes, so once again, I would like to remind all the participants, please fill up the form. It also have a column for feedback. So we need your valuable feedback. We need to improvise, we need to strengthen, and we need to go step by step and strengthen our program. So please fill up the form, and those who fill up the form, we will provide certificates of participation to them. And please give a valuable feedback. Ekach, over to you.
I would request Dr. Suvro to please introduce the next week's speakers.
So, hello everyone. The next week speaker will be Dr. Punit Babu, who is currently SR in AIMS Jodhpur, and he will be presenting on hypocalcemia in traumatic coagulopathy. And second one is the Dr. Athira from Jubilee Mission, Tricur, Kerala, and she will be presenting on Placid trial. So the time is on 14th of October and from 3:30 p.m. onwards. Thank you.
So just a reminder, next Wednesday, Placid trial is going to undergo IFTM scrutiny. We will see what they did, whether it was right or not. We would thank Dr. Debashsish, Dr. Neelam Marwaha, and Dr. Vijay Kumawat for taking time out. Sir, for guiding us all the way. Ma'am, for always being the mentor to all our actions and guide to all our steps. And thank you, Dr. Vijay Kumawat, for such a comprehensive and wonderful presentation. If you have any doubts or queries for all the participants, we at Young Professional Forum, ypf.istm@gmail.com. We are always listening to you. Please give us the feedback. Go to the chat section, fill up the form, give us feedback, and we'll give you a certificate for it. You will get the certificate for the webinar. Thank you so much. The meeting is going to end now.
Thank you. And I thank all the participants for this thing. And we again catch you all